Great, thanks very much. So I should say that Xander's basically just covered my first slide entirely. So yeah, I was just going to introduce myself, but Xander's basically covered everything I wanted to say. So yeah, I did my master's in mathematical physics at the University of Liverpool. And one thing that always makes me laugh is that whenever you see a photo, you Google image University of Liverpool, you see this beautiful red brick building. But as we all know, maths buildings are the ugliest buildings in the whole world, except this one. Except for this one. And I was just saying to you, Xander, wasn't I, actually, that it's a surprisingly nice maths building. But if you stand in the spot where this photo was taken and do a 180-degree turn, you actually see the University of Liverpool maths building, which is a lot less nice looking. And then, yeah, I did my PhD in maths at the University of Exeter. It was a very applied PhD, and I was actually based in the Living Systems Institute, where I was modelling brain networks in Alzheimer's disease. And then, yeah, I've just been doing research ever since then at Kubrick, and then a brief stint in Birmingham over COVID, so I didn't actually leave Cardiff. I barely ever went to Birmingham because no one was allowed out of their house anyway. And then back to Cardiff again. So my research involves multiscale modelling and applying methods from maths and physics to brain networks. So why is this of interest? Well, this is a quote that I really like. There are 100 billion stars in the Milky Way galaxy, roughly the same as the number of neurons in our brain. You may have to travel 24 trillion miles to the first star outside our solar system to find an object as complex as what is sitting on your shoulders. So why is the brain so complex? Well, one of the reasons that I'm very interested in is that it is a very complicated hierarchical network. So first of all, you have, I'll see if I can use this. It's probably easier just to use my mouse. Can you see my mouse? Yeah. So first of all, you have single neurons, and these neurons, their dynamics are generated by a large group of ion channels opening and closing, working together to perform a task. And then you have groups of neurons that have similar electrophysiological and morphological properties that all, again, function to perform a task. And so we can view this as like one node in a network of a bunch of neurons all working together. And then you have different types of neurons working together within an area of the brain. So this is sort of one of the simplest schematics of, a very simplified schematic of an area of the brain, where we have three of the main types of neurons. So you have your principal cells that actually do all the computing, and then you have excitatory interneurons that tell these principal cells wake up, turn on, and inhibitory cells that tell these principal cells to quiet down and stop activating. And so again, you can view the way that these different types of cells interact with each other as what we call a mesoscale network. And then at the large scale, you have these types of networks at different locations in the brain. So these act as segregated nodes in a network, but they also integrate together. They also work together. So for example, if I'm walking down the street, I might have here at the back of the brain, this is the area that processes vision. So I need to see where I'm going. Over here, this strip of the brain here is the motor cortex. So this will tell my brain to, or tell my feet to move, basically. Down at the bottom of the brain, underneath here, is the spatial navigation zone. So that's where I'll look for landmarks, and I will process what I'm seeing around me. And at the front of the brain, you have the executive functioning. This is how I plan my route, plan where I want to go. And all of these things have to work together for a simple task like walking down the street. So this is a much larger scale whole brain network. And so my research basically involves bringing people in to Kubrick, where we do brain scanning. So things, structural brain scans, like an MRI, where you're basically taking a photo of the brain. But more interestingly for me, we do functional brain scans, where essentially what we're trying to do is we're trying to measure the electrical activity of the brain over time. And this is an example of an MEG scanner. So that stands for magnetoencephalography. I also work with EEG, which is electroencephalography. But in both cases, what these do is when you have a bunch of neurons all firing together, there's electrical currents flowing up and down the cells. And as anyone with a physics background will know, if a current's flowing, you have electric and magnetic fields generated that conduct onto the scalp, and we can record them. And then essentially, the aim of my research is, once I've got this data, I do various approaches to try and analyze the data, things like looking at mapping activation, looking at synchrony between regions of the brain, trying to segment the data into different patterns of activation and connectivity that we call microstates, and then building computational models to try and explain the various phenomena that we find in this data analysis. And then I also combine this with things like cognitive testing and motor testing, various different measures of behavior and function, and all of this put together to try and help understand disorders like Alzheimer's disease, epilepsy, schizophrenia, and genetic disorders. So this talk, I'll give a little bit of background, first of all, to how we go about modeling these brain dynamics, and then I'll touch on some of the problems that we're facing with our current project that is a very interesting multiscale project. So, as mentioned, the brain is a very multiscaled network operating at various different spatial scales, and we can record the activity at all these different spatial scales. From the very smallest scale, where we might use something like patch clamp, and essentially what you do there, you've got your cell body, and you get a little pipette, and basically you look under a microscope, and you put this pipette against the cell body, you then break into the cell, and then this lets you put currents in and out of the cell. And this is an example of a patch clamp that I did during my PhD. And you can see here that I injected different levels of currents, and what you see is that the neurons don't fire, don't fire, don't fire, and then suddenly they start what we call spiking, or firing action potentials. And this is just a figure from my thesis. It's not really relevant, but really the aim here was to highlight that this is a bifurcation happening in a real system in real life. And we can model these types of dynamics either from a biophysical perspective, but also very simply just using bifurcation type normal form models. So that's measuring it at the smallest scale, and that shows it's obviously very invasive. We have to be able to isolate individual neurons, all the way up to whole scale, like whole brain imaging, where in a human, for example, the brain actually has to be inside a head. And as mentioned, we put the electrodes on the scalp. And once we start measuring at the larger scales, what you see is these synchronized oscillations occurring. So it's no longer binary spiking ons and offs. Now we have oscillations at various frequencies, and it's more of an analog rate and frequency code that is happening. And we have things like phase coherence between oscillations at different areas of the brain. So modeling any one of these scales, and I should say we can do measurements at all the intermediate scales as well. I was just trying to show the smallest up to the largest. And modeling any one of these scales is not a major challenge. It's something that we've been doing for a long time. So for example, the single cell model, if any of you have done a mathematical biology or even taught a mathematical biology course, you've probably come across the Hodgkin-Huxley model. And we can look at the biology of what generates these action potentials. For example, we can say, well, you've got a cell membrane here, and you've got ion channels, and you've got ions that have different charges inside and outside of the cell. And we see that as the spike starts to happen, some positive cell ions flow into the cell, and this causes other ion channels to open, which causes ions to flow out, and eventually you return back. And so viewing this as a resistor-capacitor circuit, where the cell membrane is a capacitor and these ion channels are variable resistors, we can build simple models like this one, and then you can just get quite rich dynamics out. And even just a simple two-current model like this one, you can get some nice spiking activity out. You can get bistability. And this bistability is particularly interesting because we can start to introduce newer currents, slow currents, for example, that might increase when you're in quiescence, when you're not spiking, until you hit this bifurcation, at which point you pop up to the spiking regime, and then this current might decrease, and you drop back down. And so this gives you things like bursting. So this is single-cell modeling, and actually this is what a lot of my very earliest stages of my career was doing. This is my very first paper, where we were interested in the bursting activity of neurons in the spatial navigation systems of the brain. And so these neurons are known to fire in bursts, and then in a rodent model of Alzheimer's disease, this bursting breaks down. And we know that people with Alzheimer's disease often have this where am I type response, and spatial navigation is one of the earliest things affected. So we built a mathematical model of these cells. We did things like analyzing the slow currents that generate these bursts, looking at the period doubling cascade that led to the bursting from tonic spiking and so on, to really try and explain why this spiking might break down, this bursting might break down. So it's not super relevant to the talk, but it's not often I get to go back to my first papers. So it's just an example of how we can do these types of analyses and understand the dynamics of disease even at the single cell level. But then when we want to go up to larger scales, for example, the whole brain, we don't typically just wire up a bunch of these neurons. That's not to say it's impossible to do if you've got a powerful enough computer, as this paper has shown, where they simulated several million neurons over 6 layers with multiple compartments across the whole brain. But that is very, very computationally expensive and very difficult to actually understand what is going on. So this paper always makes me smile. It makes me think of the Jurassic Park quote, "Your scientists were so preoccupied with whether or not they could, they never stopped to think if they should." But generally, if we're interested in larger brain, like whole brain stuff, like these neuronal oscillations, we're often interested in the phase coupling between different areas of the brain. So a more standard approach is to use oscillator-type models, models that can generate simple oscillations. So that's one simple way that we can model these large brain stuff. And on the whole brain, another application that I'm very interested in is epilepsy. So if we look at an EEG scan, so these are the electrodes placed on the scalp, and we look at an EEG scan of someone in background, these are those oscillations you saw before, but as you can see, they look quite small now because of the scaling. And as a person transitions into a seizure, their activity looks like this, where you have a lot of synchronous activity, and you have these high amplitude oscillations. So again, we've used a range of models, things like Kuramoto models of phase locking, theta models of the saddle node on the invariant circle, to try and model this activity. And so what do we actually gain from doing this? Because it's a very simple oscillator model. Why would we even bother to do this? So one approach is to gain mechanistic insight, and also it can help us develop biomarkers. So an approach that we developed, myself and many others, it was really driven by my PhD supervisor, so I was part of this, but didn't really drive it. But essentially we use a method called the brain network ictogenicity, where essentially we take our EEG, we take our connectivity matrices, we generate connectivity matrices from our brain scan, and we treat each area of the brain as one of our oscillators, our two-state oscillators. And we wire them up in such a way that the phase locking matches what we see in the data. And then, essentially, what we do is we can change parameters like the excitability, for example, in our saddle node model, and we can quantify how much time at a given excitability value is spent in the seizure state, so the percentage of time in a seizure state. And as you increase excitability, you see that the amount of time in a seizure state increases. And we call the area under this curve the brain network ictogenicity. And there's been a number of applications that have shown that this is quite useful in the field of epilepsy. So, for example, one major issue in epilepsy is actually getting a diagnosis. It can take people up to 10 years to be diagnosed with epilepsy. And one of the big problems is when you have one of these EEGs, in order to have a diagnosis, you have to have epileptic activity appear on the EEG. And so lots of people, they'll go in, they'll have their 20-minute EEG scan, and nothing will show up. And actually, epileptic activity is rare. It's more likely than not that they just didn't have anything at that time. So a big bit of work that we've been doing is essentially using this brain network ictogenicity. We've been looking at clinically uninformative EEGs, so EEGs that don't have any epileptic activity in. And we've calculated this brain network ictogenicity, and we've shown it actually does quite a nice job of separating controls from people with epilepsy. And in this paper here that we published in 2023, we showed that across 5 different hospitals of real patients who thought they might have epilepsy, it actually works pretty nicely. And now this is being used in Wolverhampton Hospital and as a trial period. So it's got real translational applications. And actually, there's just been a paper earlier this year released about some of the very preliminary results of that. Another potential clinical application is informing resection sites for epilepsy surgery. So if someone's got epilepsy and they're not responding to drugs, they will then have surgery. And so again, it's been shown in a few papers this sort of approach can help inform where you might want to do the surgery by removing a node from the model and then recalculating the percentage of time spent in a seizure. And we can basically see then how much does this brain network ictogenicity change. And something that I'm very interested in, as I've mentioned, I'm very interested in Alzheimer's disease. And one of the key things that isn't well understood in Alzheimer's disease is why people with Alzheimer's disease are more likely to develop seizures. It's quite a high portion of people with Alzheimer's disease develop seizures. And we also know that their brain network structure changes in Alzheimer's disease. So we hypothesized that the brain network ictogenicity would be higher in people with Alzheimer's disease. And that's what we found here. So for any given level of excitability, the people with Alzheimer's disease were tending to spend a lot more time in the seizure state than the controls. So this is just an example of how even the very simple oscillator models can give us a lot of insight into both mechanisms and biomarkers of disease.
Quick question.
Yeah.
I assume this is very dependent on the geometry of the network.
Yeah.
So could you tell me a bit more about how you form that?
Yeah, absolutely. So it depends on the specific application, but typically what we do is we take some EEG, so you've got sensors at different locations, and then so often we'll do something like source localization, which I haven't touched on here, but it's sort of the first step here where we build a model of the head and then try and predict where in the brain this activity is coming from, but also you can do it in sensor space as well. And then essentially we look at how phase-locked the activity at different locations are, and we use that as sort of a surrogate for how connected different areas of the brain are.
So two signals are similar, you're saying they have a strong connection?
Exactly that, and if two similar, if they're not similar, we say there's a weak connection.
And how variable is this from person to person?
The connectivity or the...
Yeah. Is there sort of a general map which everyone on, and then you strengthen and weaken?
Yeah, that's pretty much it. Yeah, there are very robust networks in the brain. They've been very well identified, and as you said, it's just different people might have different certain areas of sight. Yeah, exactly. So, yeah, of course.
When you say computational model,
Yeah.
does it work for...
So the specifics about this one is we use this model here in that paper. So essentially it's a model of a saddle node on the invariant circle. It's the normal form of this bifurcation. And so essentially in the quiescent state, you've got a stable, steady state. You can still get epileptic spikes. If the activity comes over here, you'll move around until you return back to that state. But then as the input increases, or in fact, this is the excitability parameter that we're changing here, you get closer and closer to the saddle node occurring, and then at that point, you're just spending your whole time in the seizure state. But it's the input from the other areas that sometimes drives you into going into this spiking state.
How close is that to the Hodgkin capacity? How many equations?
This is one equation. Yeah, this is one equation per reason. Yeah, so it very much is not derived from the Hodgkin-Huxley, and actually that was the point of my next slide. Is that that's very much a phenomenological model. It's very much it does two things, like a seizure or like not a seizure. And one of the major questions, both in the modeling literature but also in the experimental literature, is bridging the gap between these. You know, how does perturbations to the spiking of the single cell affect the oscillations and vice versa? And so there's sort of a major challenge to this. And this is one of the reasons that I think for understanding the brain, modeling is so important, because I think the models can really help us understand these interactions between scales in a way that potentially experiments can't, and models and experiments can potentially inform each other. So let's move on to trying to bridge the gap with some of the models that we're using in our current project, and then I'll get on to how we're applying this in the current project. So let's try and go from a single cell to a bunch of neurons of the same type all working together. And what we do is we use something called a neural mass model, where we model all these cells working together as a single mass. And one approach that we use specifically for trying to bridge the gap, there's loads of different groups of neural mass models out there, but for trying to bridge this gap, we can use a type called mean field models. And essentially, mean field models, we start off with the equations for a neuron. So it can be our Hodgkin-Huxley, it can be our resistor-capacitor type neurons. And then we assume we've got a load of these neurons, and each neuron can be represented by a point in our n-dimensional space, where n is the number of equations, that has a certain velocity. And then we can use things like the Fokker-Planck equation from mean field theory to try and basically we assume there's some distribution of the, their location follows some distribution, and then we can try and derive equations for the dynamics of the mean and the dynamics of the dispersion. And I will say, in practice, what we almost always do is we assume that the dispersion remains constant, and we just follow the dynamics of the mean. So this term tends to go to 0. And obviously, this is not a very easy thing to derive, and so we make a lot of assumptions about the distributions and so on. But there's a few common ones out there that are quite used, and probably the most widely used of these types of mean field model is the conductance model, where we start off exactly as I said. We start off with a simplified version of this resistor-capacitor circuit, where basically it's the exact same as the Hodgkin-Huxley, but we get rid of the sodium current that causes the spikes, and we get rid of the potassium current that causes the reset, and instead we call it an integrate and fire model, where basically we say once we reach some threshold where this sodium current would turn on, we just assume there's an instantaneous spike and we reset it. And then essentially you can follow through the Fokker-Planck dynamics. We make some assumption that the overall firing rate based on this, if we assume that the data is Gaussian distributed, then essentially we know that the inverse of the cumulative distribution function for this will be a sigmoid. So we assume that the firing rate for any given mean voltage will follow a sigmoid function. And then you can essentially derive a mean field model with this as the firing rate. So that's one common approach. More recently, over the past sort of 5 years or so, a new model that has been nicknamed the next generation neural mass model has been developed. And we start now with the quadratic integrate and fire neuron. So instead of just having this linear model, we assume it actually follows this bifurcation structure. So you have your resting potential and your threshold potential, and as you increase the current, your voltage goes up to infinity and then resets back to negative infinity. So we can write that like this, or equivalently, and it's a very simple transformation of variables, we can write this in phase space like this. And those are the exact same model, and this again is that same model that we've spoken about before. And one of the cool things about this model, the fact that you can write it in two different ways, means that we can follow the Fokker-Planck equations that I spoke about before for the voltage. And this, what you derive, is an equation for the average membrane potential and the firing rate. And under a bunch of assumptions, this is actually an exact description for the spiking neurons, which is really cool. But also in this phase space, where you have a phase oscillator that is sinusoidally coupled, you can use the Ott-Antonsen ansatz that was developed in 2008. And this gives us an exact description of the Kuramoto ordo parameter, so the average phase and the within population synchrony. And what's really interesting is that if you compare these two models, you can directly transform from one to the other. So these are the exact same model, even though they're derived in two separate ways, which means that basically I can simulate either one of those two models, and I can calculate the other. So I can simulate the voltage and firing rate of this population, and then I can work out the average phase and the synchrony within the population. And this is going to be important later, and this is one of the reasons that we're using this model now. So now we've got a model of a neural mass. It's fairly simple to extrapolate this to a cortical column. We have to pick some layout for the cortical column, and of course that is a choice that you have to make when designing your model for your experiment. You can go from very simple, where you've just got excitatory and inhibitory neurons, all the way up to the extremely complex. And as with any model, there's a trade-off between how complex you make it in terms of things like identifiability, overfitting, interpretability, and the simplest, you might miss important mechanisms, you might get unrealistic responses. And the way that we wire these up is we use models of the synapse. So for that, we use a differential operator. There are other models out there, but the most common we use a differential equation that looks like this. And the reason we model it like this is that we can then directly model the response to a single spike, the impulse response, using a Green's function. So if we have a single spike, the equation becomes this, and then two of the most common Green functions we use are these ones. And you can see that they match the data quite nicely. So we can directly model the response to a single spike, and then we can basically just use this equation to convolve these with every spike that is happening. And that's how we model the connections between these regions. And at the same way, we can then model these cortical columns. We can wire them up together. And here's just an example that is from a common experiment we do. It's not really important, but it just demonstrates the point that you have your cortical column, one cortical column at each of these 5 locations for modeling this particular model, and you can wire them up. And so now we've basically managed to go from the smallest scale all the way up to building a model of the whole brain, or a subset of regions of the whole brain, where we've still got parameters in our model that are directly interpretable in terms of electrophysiological of the single cell. So the current project that I'm working on now is modeling multiscale dynamics in genetic risk of schizophrenia. And so this is called the Converge project. So we're very interested in these, what we call copy number variants, which are genetic mutations that are either additions or duplications of certain sections of DNA. And several of these copy number variants give genetic risk of schizophrenia and psychosis. These same ones also give risk of autism, ADHD, low IQ, but we've picked specific ones that give us risk of schizophrenia. And so this project, well, actually, I should say that when the project was originally applied for, it only had 3 aims. So aim 1 was to bring the kids in to our MEG scanner, and we can record the large-scale oscillatory activity, and we can try and map activation patterns and map these connectivity patterns that we've spoken about before. So here we're really looking at the large-scale oscillatory stuff. Aim 2 was to have rodent models where they have the same copy number variants as the children that we're bringing in. And in these cases, what you can do is you can stick electrodes down different areas of the brain, and you can look at, for example, the activity at different layers of the cortical columns. And again, that sort of gives us a mesoscale. And then aim 3, what we do, and I keep saying we, I do this aim 1. It's a very clever group in Bristol that does the group for aim 2, and a group that, this next step is basically magic to me, and they do it in the Hayden Ellis building in the medical school. But what they do is we collect bloods from the kids when they come in, and they convert these bloods into stem cells, and then they convert the stem cells into neurons. And so then they can grow basically 150,000 neurons in a Petri dish, and then they can either do this patch clamping where they measure the electrical properties of single cells, or they can look at how these neurons all interact together in a multi-electrode array where you have a grid of electrodes and you can look at the spiking activity. So this gives us a lot more microscale information as well.
Do you know that the neuro cells that you derive from there will be equivalent in some way to those in the brain?
So then they are equivalent to very early developmental ones in the brain. So, for example, in my other passion, which is Alzheimer's disease, we almost definitely would never use these because the things like the epigenetics are super important there, where things like smoking can affect how your DNA activates and so. And so this can affect. So basically, Alzheimer's occurs in a very old age. So using these is unlikely to give us a lot of information. Whereas in this, because it is a genetic disorder, it's directly in their genes from the day of, conception, I suppose. And so, yeah, these aren't necessarily exactly the same as what's going on in the brain, but we can...
The DNA's in the instructions.
Exactly. And so things like the time constant of these neurons, we might not say that it matches exactly what is in the large scale, but if there is a difference in the time constant across groups, it's likely that there's also going to be a difference in time constant across groups in the larger scale activity as well, if that makes sense. And when they originally applied for this, there was only those 3 aims. And actually the application got rejected, and they were told, this is 3 separate projects, how are you going to bring them together? And so that's where the computational modelling comes in to try and bridge the activity at these scales. And so I do AIM 1, and I'm doing AIM 4. And I don't expect you to know what all these different experiments are, and I've not got time to go over them all. But what I really want to highlight is that between the various experiments we do in the MEG scanner, between the cell lines, between the rodent activity, we can really probe pretty much all of the different scales that I've spoken about now with various different experiments. And we've just sort of got to the stage now where we've got quite a large data set. I've spent the last 2.5 years collecting data for this. And so we start in with the model development now and really trying to start modelling this. And so I've highlighted this 40 Hertz auditory steady state response. This is where we've decided to start with the modelling. And one of the reasons is it's at a nice mesoscale activity. In fact, you can see that it doesn't even necessarily recruit the entire cortical column. As I'll show you in a moment, it recruits subsections of this cortical column. So it's at a nice mesoscale activity, but it's also very applicable to human and rodents. So before I go into too much detail about this, I'll just explain what this task is. They're in the MEG scanner, sit in. They're watching a video of someone making Lego. This isn't really part of the task. It's just because we've got kids with ADHD and you can't ask them to sit still for too long without something to entertain them. And then basically we play a 40 Hertz click train for half a second at a time. So basically clicks, 20 clicks for half a second. And then, as I mentioned, you have the response to this is currents flowing, which make a magnetic field that is measured by the MEG scanner. And if we look at the responses to this, what you tend to see is that the brain activity will generate these phase-locked 40 Hertz oscillations that are locked to the stimulus, and it's basically an increase in the 40 Hertz power. And in people with schizophrenia and psychosis, it has been very robustly reported that both the power and the phase locking of this 40 Hertz response is reduced. And another reason that we think it's a nice place to start is because it's about the only experiment that we can exactly replicate in the mice as well. And so this is an example of the 40 Hertz response in a mouse. Pretty much all of the other experiments, even something like resting, you can't really just ask a mouse to sit still with its eyes closed for 5 minutes. But this one we can do. And yeah, in both the humans and the mice, in the imaging data, we found a reduction in this 40 Hertz power. So I wanted to try and start modelling there. So we used the mean field model that I spoke about earlier, the next generation one, because we're also interested in the phase locking to the stimulus. And the next step is then choosing a model architecture. And this is actually sort of an open question that I'm quite interested in people's thoughts about. So should we try and build one big model that can capture all the different experiments that we're doing, or should we make bespoke models for different experiments? And this is just an example of a paper where they had 3 different experiments and they used the same model for it. But actually some of the parameters that they got out, to me, don't seem very meaningful for certain things. And actually when I spoke to Rick Adams, the first author of this paper, he said in their group they are starting to move more towards bespoke models. So that's the approach that I'm taking, but if people here have any thoughts on this, please do talk to me, because it's something that we're still not 100% sure on. But even if I do try and build a big model, it's kind of difficult to choose one that can capture everything. So let's take a look at this thalamocortical model that is probably the most complex model that is currently being used regularly in the literature. And one of the reasons... Yeah.
So choosing a model it shouldn't be too difficult if you know what you want to do with it.
Mm-hmm.
So what do you want to do with it?
Yeah, so
it depends. If you want to verify the same experiment as you did before, obviously the bespoke model will be the one you need.
Yeah.
But if you want to make predictions about things that you can't experiment about, then you need to throw as many experimental data into that model and hope that it will give you some. But I also have another question. Okay, so you have this group of kids. How did you select them?
So they are recruited. All the ones that we've got up to date have participated in previous studies. So they've all got a genetic.
still.
Yeah, it's, they've got a genetic diagnosis, so they see the doctor, yeah. And essentially, yes, we actually also, we verify all this, so when they come in, they spit in a tube, so we verify they have the genetic disease.
So there's a family history, but
yeah.
Do you also test kids without a family history?
Yeah,
or
we test...
kids who don't
yeah.
have all the family history?
So we don't test without the family history at the moment, but we're about to start. So right now, all of our controls are siblings, so they've got the same genes, but they don't have the genetic mutation. So essentially, we've got siblings that don't have the mutation versus the kids that do. And that's our control set at the moment. But we've actually just got ethics to start doing community controls as well. So we will also start doing that. But right now, we've not. All the results I've shown so far have only been versus siblings. So again, going to this model architecture, let's take a look at the most complex model that is currently being used commonly in the literature. And it's a thalamocortical model. So you've got a node in the thalamus, which is this part of the brain deep inside, and an area on the cortex that we would put here. And the reason we do this is because the auditory pathway, it goes up here through the brain stem, so from the ears, through the brain stem, and then the first sort of real part inside the brain is this thalamus, and then it goes to the cortex. But let's now look at the literature and see what the spiking activity actually looks like in mice. And if we look at the cortex area, they looked at spiking activity at different layers during this 40 hertz task. And what they found was actually in these superior supragranular layers, so basically layers 1, 2, and 3, there wasn't really any 40 hertz spiking going on. So we probably don't actually need that in this model. In these infragranular layers, again, not really much 40 hertz spiking going on. So those layers are probably not really that relevant to this particular paradigm. And it's a lot of complexity in the model that isn't really super important. And then when we look at, they separated this into the inhibitory neurons, which are basically the red dots, and the excitatory neurons, which are the black dots. In these granular layers, by far the most phase locked to the 40 hertz stimulus was the inhibitory neurons that aren't actually included in this model. So even though this is a super complex model, it's not that appropriate for the same thing. And similarly down here, you can look, this particular population doesn't have the 40 hertz response either. So it's not necessarily actually that useful a model. So what we've done here, and again, I'm not sold on this. I'm not like committed to this if people have other thoughts, and it's something I'm keen to chat about. But I've taken a look into the literature at the circuitry that we think generates this 40 hertz response. And so here I've only showed the right cortex, but we've also got the left auditory cortex, which is identical. And so we think it's something like this. You've got the input to the thalamus, which goes out to the inhibitory and excitatory interneurons. We've also, there's 2 time scales of excitation and inhibition that are important in the model, and particularly these act very slowly. And in fact, actually, the slower time scale is much slower than you tend to see in the 40 hertz rhythm. So we can probably assume that while they are important for stabilizing the orbit, by the time you've got a stable periodic orbit in the 40 hertz, these are probably remaining largely constant. So we've actually replaced them with a constant input. So my final model looks something like this. And we can go to the literature, we can go to some of the experiments in the microscale in vivo. So these are actually in real mice. And they have measured properties that we can use for our next generation neural mass model. Things like the input resistance, the membrane time constant, the resting threshold, the action potential threshold. So we can actually design it so that these are based on cells that have the properties that are found in the mouse auditory and thalamic cortices. And then basically the stage that I'm at now is I wanted to test my model before using it on the kids that we are going to finally make inferences on. So I took the Welsh Advanced Neuroimaging Database that's got 169 participants, healthy older adults, young adults, who did the same task. And here's what the response in left auditory cortex looks like across these 160 participants, where you can see basically it's not phase locked, and then once the clicking starts, everyone gets phase locked to pretty much the same thing. And I've started off by trying to make my model reproduce this. So one thing that I thought was really interesting is that in the data, if you compare the thalamus to the auditory cortices, you see this phase lag that is consistent between them. And we see that in our model as well, which is kind of a nice validation. People have also, then they also showed in that paper that the within groups, within population synchrony of these inhibitory cells, the fast spiking ones, was actually higher than it was in the regular spiking cells. And again, we found the same thing once we used the Kuramoto order parameter. So similar sort of idea that the fast spiking cells are the ones that are more synchronous. People have done optogenetic experiments where you can basically inject your neurons with a virus and then use lights to control whether they are firing or not. And you can see that by suppressing the fast spiking cells, you get a reduction in the 40 hertz amplitude, and that's what we see in our model. And by activating thalamic reticular cells, you also see a reduction in this 40 hertz amplitude, and that's what we see. And so that is basically the stage that I'm at now. So the plans moving forward are to start fitting to our children data and start analysing the results there. Start fitting to the 40 hertz in the rodents and seeing whether our results are consistent with humans. And then our team over in the Hayden Ellis building are going to start some new experiments with the cell lines. So we can start trying to integrate the information from those scales in terms of what might be changing at the microscale. And finally, testing the model predictions. So they're about to start doing pharmacological experiments in the cells and using depth electrodes to stimulate different areas in the rodents. So we can also, again, start to test whether our model predicts changes that can be found in the rodents and cells. And finally, at the longer term, can we build models of a larger scale auditory task? So these ones that integrate different areas of the brain and the auditory resting networks. So basically start to integrate this model in a wider model of the brain. And finally, I think I've got a couple of minutes. One thing that's kind of interesting, and again, if people have thoughts on this, I've only very briefly started thinking about this. In the cell lines, what you tend to see is that they have these bursts that are at a very slow timescale. I'm talking like one burst every minute or so. And that is way slower than any of the timescales that we actually see in the neurons. And you can build mean field models. You can treat them as one single neural mass model, and you can see bursting, but it's at the timescale of several a second, which is what we expect from the timescale of neurons. So I'm starting to wonder whether it's something to do with the spatio-temporal property, and modelling multiple ones of these on a 2D grid might give us some of this more spontaneous bursting activity, but I don't know, and the biologists don't really seem to know where this bursting activity comes from either. So that's, again, another potentially interesting mathematical problem in the longer term. So, thank you. That's my talk. That's sort of where I am now. These are some of my collaborators on the project. If you have any thoughts on any questions for me now, or any thoughts on any of the stuff that I've mentioned, please do let me know. Yeah.